Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen at any time. The questions that you type there will only be visible to myself and our speakers. So thank you for joining us today. It is now my pleasure to introduce our speaker, Gareth Rogers from Refine and Kirsty McManus from Pharmaron.
Hi everybody, my name is Gareth Rogers and I am a product manager at Refine. Thank you for joining Kirsty and I today for the Ask the Expert webcast hosted by BioProcess International. In terms of the agenda for the short presentation today, I'll start off by giving a very brief introduction to mass photometry. I'll then move on to talking about the SAMX MP mass photometry system as a solution for AAV analytics. And I'll then pass you over to Kirsty, who will give you a brief introduction to Pharmaron and the capabilities and services that they can offer to their customers. And Kirsty will then wrap things up by focusing on the data that Pharmaron captured during their recent testing of the SAMX MP instrument. So to kick things off, we'll start with just a quick poll question. So on your screen now, you should see poll question 1. Which technique do you predominantly use for AAV analytics currently? And you'll have a number of options which you can click with your mouse. So A, AUC, B, mass photometry, C, cryo-TEM, D, ELISA qPCR, E, AEX HPLC, F, UV vis detection combined with SLS or DLS, G, SECMALS, or H, mass spectrometry. I'll just give you a couple of seconds to answer that question and then we'll get underway. Fantastic. Thank you. Right, so I believe most of the audience will be familiar with viral vectors as tools for delivery of a DNA payload. And that is indeed what AAVs can be used for. In addition to full capsids that contain the desired payload, manufacturing processes currently can also lead to a large number of empty capsids that are produced as well. And this is something that we need to pay particular attention to, because high levels of empty capsids can pose a number of risks. So the low transduction efficiency due to receptor binding competition, for example, can lead to an increased immune response, limited target genome concentration due to capsid precipitation, as well as an increased cost of goods as we strive to purify and enrich for more full capsids. So we believe that one of the reasons behind a large number of empty capsids is as a consequence of not having the right analytical tools for the quick and easy assessment of the empty to full ratio. So in our opinion, the ideal analytical solution would be able to accurately measure empty to full ratio within minutes. Ideally, it would be able to quantify partially filled particles. It should be able to work with low sample volumes and at a low concentration. Should be very easy to use and with a low operational cost. Serotype agnostic and applicable at different purification stages throughout the process. And we believe that mass photometry is a solution that could potentially fill this gap. So what is mass photometry? Essentially, it's all about using light to weigh molecules. And the working principle is very simple. Molecules scatter light, and we can measure this scattering. So what you can see on the top left here is a glass cover slip. And as we shine laser light towards that surface, as shown in the example here, AAV particles bind to the surface, this light is then scattered. More specifically, it is the interferometric analysis of the scattered signal from the AAV particles in this case that allows us to determine their mass. In the middle image here, where you can see it labeled as MP signal, this just shows that we capture a series of images using a camera. And three components contribute to image formation. So the light that is reflected from the glass-water interface, and the light that is scattered by the AAV particles, in this case, as well as the interference between the light that is reflected and the light that is scattered. And this interferometric signal is proportional to mass, and if we use a calibrant of known mass, that allows us to extract the mass of our sample. On the bottom panel here, that just shows that every single event that is detected is counted and a histogram is generated showing the mass distribution of biomolecules or particles such as AAVs in solution. And we believe that looking at mass could be particularly useful for AAVs, primarily because of the large mass difference between empty AAVs and full AAVs. So in a typical AAV prep, we would expect to see contaminants at a mass of under 2 megadaltons, empty AAVs around 3.7 megadaltons, and for full AAVs, dependent on the DNA payload, a mass of 4.1 to 5.3 megadaltons. On the bottom here, you can see some mass photometry data, and we can clearly see that the empty and full AAV peaks are nicely separated. This is the SAMX-MP, and this is something that we officially launched in January of this year. This is a mass photometry system that we specifically developed and optimized for AAV characterization. And this technology is optimized for the mass range that is relevant for AAVs, 3 to 6 megadaltons. It's also optimized for facilitating decontamination as well, based upon the selection of materials used in the construction of the instrument. The software is also optimized for a quick and easy assessment of empty to full ratios. We know that the SAMX-MP performs consistently across the entire empty full range. So shown on the left here is mass photometry data that we captured in our laboratories in Oxford, where we used 5 different mixtures of empty and full particles, ranging from 0 to 95% full. On the x-axis, we can see the predicted loading, and on the y-axis, we actually have the measured values captured with SAMX. What we can see is that we can reliably measure throughout the empty to full range. One of our first collaboration partners, Cell and Gene Therapy Catapult, performed a very similar experiment. In this case, they used empty and full preps, where in this case, full was 70%, and they mixed these in different ratios. Again, on the x-axis, we can see the theoretical values, and on the y-axis, the values captured using the SAMX instrument. And as we can see, there is a very good agreement between the two. The variability that we can see in the center there just relates to mixing of the sample rather than being due to the instrument itself. Nevertheless, just to be sure of that, they then worked with a prep that contained a full counted percentage of 50%. And in this particular experiment series that you can see on the slide here, they performed a measurement of 6 independent replicates in 6 independent runs performed by 2 different operators. And the criteria that they were looking to meet when assessing the SAMX instrument, you can see their acceptance criteria on the left-hand side of the table. So they were after a repeatability with a coefficient of variation of less than or equal to 20%, and an intermediate precision with a CV of less than or equal to 30%. And the qualification results, and this is actually the data that they generated using the SAMX, we can see that this is much better. We've exceeded the criteria, their acceptance criteria. So repeatability of 10.87%, and for intermediate precision, the CV was just under 12%. Is the SAMX-MP agnostic to different AAV serotypes? Based upon our data and data from our collaboration partners, we can confirm that is the case. On the left, you can see a series of measurements for 4 different serotypes that were carried out in our own laboratory over here in Oxford in the UK. And we can see that the profiles for each serotype are very similar, and each sample was handled in exactly the same way. Another one of our collaboration partners, Utrecht University, they performed a similar experiment looking at a couple of different serotypes, 2 AAV8 samples from 2 different commercial suppliers, as well as an AAV5. And their results agreed with ours. There is no difference between different serotypes. This particular lab, which is run by Professor Albert Heck, also has considerable expertise in charge detection, mass spec, and they then compared the mass of one of the AAV8 samples captured using CDMS and compared that to the data that they'd generated using the SAMX-MP. And what you can see is the 3.83 megadalton value generated by the CDMS instrument is in very good agreement with the 3.81 megadalton recording that was reported for the AAV8 Company B sample with the SAMX. How does it compare to the gold standards of AAV analytics, AUC and cryo-TEM? This was a benchmarking study that was performed by Catapult, and the SAMX-MP data agrees very nicely with the cryo-TEM and AUC data across the empty to full range, from samples that contained a low percentage full through to samples containing a higher percentage full. So SAMX-MP can reliably measure empty to full ratios for AAVs. Very limited sample preparation is required, and mass photometry is a label-free measurement where we can work with samples in their native state. You can capture results very quickly, typically less than 5 minutes, and that includes the analysis step. You can use a low amount of sample, typically 10 to 20 microliters. It's very easy to use, has a small footprint as it is a benchtop instrument, and the low operational costs are also another appealing benefit with this system, with each individual measurement typically costing less than $5. So with that, I'll move on to poll question 2 before I hand you over to Kirsty. So on your screen now, you should see the following question. After empty to full ratio, what is the next most important piece of information that you would like to capture from your samples? A, physical capsid titer, B, vector genome titer, C, vector genome size, or D, presence absence of aggregates. So if you select which one you would be most interested in finding out more information about, I'll just give you a few seconds before I pass you over to Kirsty. Fantastic. Over to you, Kirsty.
Hi, thank you, Gareth. I'm Kirsty McManus, a senior scientist within the Analytical Sciences Department at Pharmaron in Liverpool. So as Gareth said, I'm going to give you a brief introduction to Pharmaron before moving on and going through some of the data that we generated as part of our beta testing study of the SAMX-MP. So our mission at Pharmaron is to support our partners' success in the discovery, development, and commercialization of innovative medicines. We have over 15,000 employees across 3 continents, and all top 20 of the global pharmaceutical companies are our customers. So at Pharmaron, we focus on adeno-associated virus gene therapy strategies, and we have the platform to produce multiple AAVs. We have strong expertise in biologics, which has allowed us to develop AAV processes with the end in mind and supply the necessary critical starting materials. We were able to leverage an extensive collaboration network, which has allowed us to continuously improve our processes and analytics and maintain our leading position in the industry. We have access to robotics and automation, which allows us to increase process understanding, accelerate CMC timelines, and also reduce the cost of development. And we have a multi-AAV HEC 293 suspension-based triple transfection and a chromatography-based purification process. And this is driven by single-use technologies that allow us to ensure scalability and flexibility up to the 500 litres scale. So underpinning our expertise, we have custom-built state-of-the-art facilities and cutting-edge analytical technologies. We have many years of experience working with viral gene therapy vectors, recombinant proteins, and antibodies. So Pharmaron is investing in an ambitious growth strategy, and we've committed to a multi-million investment in the site, where we have a significant expansion space available for further development, clinical manufacturing, and future commercial manufacturing capacity. So AAVs consist of a single-stranded DNA genome, which is encapsulated by a protein shell, which is known as the capsid. Wild-type AAVs are engineered to produce recombinant AAV vectors that are used in gene therapy. So to do this involves the removal of the rep and cap genes of the viral genome and the insertion of a transgene to replace them. And it's this transgene which provides us with the therapeutic effect. So therefore, it's critical that we have robust characterization methods that allow us to accurately quantify the proportion of AAV capsids that contain our therapeutic transgene. So in our experience at Pharmaron, the empty full analysis is one of the biggest challenges in AAV. The methods we use for this need to be robust, they need to be reliable, they need low material costs in terms of volume and concentration, and they need to be quick. We need all of these kind of qualities in a method in order to enable us to support process development and facilitate decision making in this process. So now I'm going to move on and demonstrate to you some of the data that we developed at Pharmaron as part of our beta test of the SAMX mass photometer. So we were really excited to have the opportunity to use the SAMX mass photometer. We saw some promising data from Refine in terms of we had a much improved resolution for AAVs from the previous models of the mass photometer. And the hope was that this could be a disruptive technology for empty full analysis and further expand our orthogonal approach to empty full capsid ratio analysis at Pharmaron. So as part of our beta test, we assessed the following. We looked at different serotypes. We assessed the resolution and linearity between empty, partial, and full species. We did this by preparing various mixtures of empty and full standards. And we also used it to analyze process development samples as well. So I'm going to begin by showing you a comparison of some of our empty full data that we generated on the SAMX MP to the AEC for two different serotypes. So the AEC is kind of the gold standard for empty full analysis. So what we did was we took commercially available AAV material of drug substance purity and for two different serotypes and tested them on the mass photometer. So on the first serotype that we looked at, we detected empty and full species, and we've got a comparison on this slide of what we generated on the SAMX versus what we generated on the AEC, and we saw good agreement between these two technologies. We then analysed an additional serotype. So in this serotype, we were able to resolve empties from partials from fulls. And we looked at each of these species independently, as well as summing together the partials and fulls. And as you can see from the data in the table underneath, we saw really good agreement between what we saw on the SAMX MP and the AEC. So this was really encouraging that we saw similar results between two different orthogonal technologies. So in this serotype on the right, where we were able to see empties, partials, and fulls, we built on this a little further by mixing together different blends of the sample. So, so we took AAV reference full, so this was commercially available material, and AAV empty, and they were mixed in various proportions and tested on the SAMX MP. So based on the characterization of a mix of 100% of the full sample, we predicted what we'd expect to see in each of the blends. So this is the expected percentage full and the observed percentage full. And we plotted these two together in this chart, and we were really pleased to see a linear relationship with an R-squared of greater than or equal to 0.96 between the expected and observed total percentage full capsids. So as we were able to resolve the partial and the full species, we analysed these two species independently. So we looked at the percentage partial and the percentage full independently of each other and did a similar plot. So for the percentage partial, again, we saw an R² greater than or equal to 0.96. And for just the full by itself, we saw an R² of 0.89. So now I'm going to move on and show you our process development material analysis. So we took samples from a non-optimized process. As part of this process, we have a capture step followed by 2 additional polishing steps. So this data here is the data we observed for the capture step eluate. So what's eluting off of the capture step, and we saw approximately 9% full. The mass range for AAV is already fairly clean looking even at this stage. We do have some evidence of lower mass species, but it's not interfering with the empty full. It's well resolved away from that. So from the eluate of the capture step, we then prepare our sample for loading onto the polishing step. So again, this is essentially the same sample as the previous one. And again, we saw about 9% full. So this is a chromatogram for polishing step A. So what we do is we pull together fractions 5A2 up to 5B1 and pull that together as E1. Then we have pulled fraction E2 and pulled fraction E3. So the mass photometry data for each of these pulled fractions is displayed here. So in fraction E1, we had a very small amount of AAV detected, kind of as expected. In E2, which is where the AAVs elute from this step, we saw about 17% full, and this is the fraction that we load onto polishing step 2. And at this stage, we have cleared all of the low mass stuff by this first polishing step. And in pulled fraction E3, we are able to detect some AAV, but the percentage fall is lower at only around 6%. And we see mostly empty material here, which ties in nicely with what we see in the chromatogram. So the mass photometry data at this stage is confirming our chromatographic data. So fraction E2 is then loaded onto polishing step B. This is the chromatogram for polishing step B. Again, these are pulled fractions from the chromatogram underneath. In fraction E1, we saw a very small amount of AAV detected. In fraction E2, we saw approximately 0.5% full. In fraction E3, the percentage full is starting to increase. We suspect at this stage that both partial and full are eluting. And then in fraction E4, we're significantly increasing the percentage full in this fraction, and we're now about 60% full. So again, the mass photometry data confirms the chromatographic data. So in conclusion, mass photometry is a rapid, easy to use empty full capsid characterization technique, which requires low sample volumes and moderate titers with an approximate concentration of 10 to the 11 particles per mil. Mass photometry is serotype agnostic and provides similar results to orthogonal empty full capsid method by AEC. At Pharmaron, we were able to demonstrate linearity and resolution between empty, partial, and full AAV capsid species in the reference sample. So mass photometry appears to be a promising new technology to aid in the optimization of process development protocols. So at Pharmaron, we are constantly reviewing and adding methods and looking to leverage platform approaches wherever possible. One of the biggest challenges in the gene therapy landscape is measuring potency. We have capabilities in-house to develop and perform a combination of platform and product-specific assays in-house with our bioassay team. So I'm just going to end by thanking my colleague Lauren Tomlinson at Pharmaron, a scientific associate for free, because she generated and processed the process development data which I've presented today. I would also like to thank my colleague Paul Getty, our senior technical specialist at Pharmaron, for planning and coordinating our mass photometry beta testing. And finally, I would like to thank the product managers at Refine, Sveya and Gareth, as this project would not have been possible without the support of the team there. So I'm going to hand back over now to Gareth to finalise the talk with our final poll question.
Thanks, Kirsty. So the final poll question 3, for which process do you see mass photometry as being most relevant for in your future AAV workstream or workstreams? A, as an R&D tool for drug development, B, as a screening tool during process development, or C, as a quality assessment tool in final drug testing. So you can select your answer, and I'll give you a few seconds, and then I'll hand you back to Leah for running through the Q&A session. Okay, thank you. Over to you, Leah.
Okay, great. So we do have some questions from the audience. As a reminder, you can just go ahead and type those in and we will see them. So the first question is for Gareth. What is the minimum AAV sample concentration you can use with the SAMX?
So where sample concentration is concerned, in order to generate the best results, we would typically recommend working with a sample concentration of 10 to the 11 particles per mil. But you can certainly work with samples that have a particle concentration of 10 to the 10 particles per mil.
Okay. And the next question is for Kirsty. Have you run any pre-capture process development material, and was the MP data useful?
Yeah, we have. We have run some harvest and some clarified material. For the harvest, the signal from the AAV wasn't strong enough to be distinguishable from the impurity in background, but for the clarified material, we were able to generate a result despite the signal from the low molecular weight impurities. But I would say that from a process development perspective, in my experience, the system really shows its value in the optimization of kind of DSP protocols rather than USP, but we are able to generate some useful data for USP, certainly.
Yep. And for Gareth, can the SAMX be used for analyzing other viruses?
Yeah, there's certainly nothing to prevent you from looking at other viral samples. It just primarily comes down to the diameter, the size of the virus. So the absolute maximal detection limit, I'd say, would be nearing an 80 nanometer diameter. Certainly for viruses that have a diameter similar to AAVs of about 27 nanometers, that's completely fine. So yes, is the answer.
And for Kirsty, based on what to define the boundaries of E empty, P partial, and F full?
Okay, so empty and full is quite straightforward, really, because we know what the mass of an empty particle is, and we know what the size of our transgene is, so we kind of know what we're expecting to see for those two particular ones. The partial is a bit more challenging, but as we saw resolution between the three species, we could kind of see where the peaks and troughs were, and we defined the boundaries kind of based on that.
Okay. And for Gareth, what is the difference between the AAV mode on 1MP and SAMX?
That's a very good question. So having become a new member to the Refine team, I have not worked with a 1MP myself. So if that person has their contact details, I can ask somebody that has the relevant experience to provide you with a more detailed answer than I can.
Yes, we'll pass that on to you. Okay. And for Kirsty, what's the biggest benefit of the SAMX MP versus your current technology?
So in my experience, the biggest challenge in AAV analytics for empty full analysis, it's kind of a combination of time and material requirements. And I feel like the SAMX MP is able to help with both of these biggest challenges. For example, just as a comparison to kind of the gold standard AUC, we require 400 microliters of material at kind of E to the 12 concentration, and our kind of throughput is only around 7 samples a day with kind of an overnight run. And sample processing can also be time consuming as well. With the mass photometer, the SAMX MP can kind of deliver a single result in just minutes using only 10 microliters of material at kind of 10 to the 12. And also we're able to kind of generate multiple replicates with a kind of rapid turnaround. So it's really, really useful in terms of reducing the amount of time spent and the amount of material spent.
Okay. For Gareth, what is the throughput of the mass photometry instrument?
So with the instrument that we've looked at today, typically you would be working with some consumables that have a series of wells inside them, and it would be inherently manual. So you would load samples sequentially, one at a time, but you can work through many samples quite quickly. It's just a case, as I mentioned during my talk, where you load the sample and in under 5 minutes, including the analysis step, you get data. So it's just as inherently manual, but you can look through as many samples as you want to, as you have available quite quickly.
Okay. And for Kirsty, how much time does it take to quantify your AAVs along your PD process using MP versus AUC?
Okay, so because we only had the instrument for a limited amount of time, we haven't kind of really had the chance yet to kind of put it into process kind of live as we're kind of developing our PD process. We've kind of just looked at it kind of at some samples that we already had as we were kind of already developed that process, just to see whether it would be useful. We have got a mass photometer kind of purchase going through this year, so I will know more about that later. But typically, as I said, we've only got, for the mass photometer, we're able to get a result in minutes, but kind of for the AUC, it takes kind of an overnight run. So it is a significant improvement in the time taken.
Okay, so it looks like we have time for just one more question. If we didn't get to your questions, and there are a few of those, we'll be passing those along to Gareth and Kirsty to follow up with you directly. But the last question for the webcast is, what consumables are required for a typical measurement? And that's a question for Gareth.
Yeah, so in terms of the consumables, typically it'd be a glass cover slip with a what we call a sample well cassette, which is essentially kind of like a silicon gasket containing a series of wells. And these are all consumables that we have made available for our customers to purchase directly from our website. So we've recently established a web store where customers and end users can go and buy these consumables directly. So with the idea being that that would reduce their sample preparation time, so they can start capturing data much more quickly.
Okay. Well, thank you to Gareth and Kirsty, and thank you to our audience for joining us. The recorded version of this webcast will be made available for on-demand viewing on our website. As a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.